All right, hello everybody, and welcome to our presentation. Next slide, Vivian. So we're going to be presenting about our work with the US FDA, and I'm going to be describing what part of FDA we're working with and what this contract's under. But first, let me just point out the team, which comes from Columbia University, Northeastern University, University of California, Los Angeles, and Johns Hopkins University. And I will, in a few slides, be talking about the entire Odyssey team that's working on it, which is beyond this group. Next slide. So we're working with the US Food and Drug Administration, specifically with CBER, the Center for Biologics Evaluation and Research. And their mission is to enhance public health about biological products, including blood, vaccines, allergenics, tissues, and cellular and gene therapies. So we worked with this group in the past. IQVIA had a contract for several years preceding this, and Odyssey was part of that, helping learn about, for example, transfusion medicine. It was part of the BEST initiative. So this is part of CBER, Biologics Effectiveness and Safety System, which is to access new and better data sources, methods, tools, expertise, and infrastructure to conduct surveillance and epidemiologic studies. That started in 2017, so we were part of it from the beginning as part of the IQVIA contract. Okay. The vision of BEST is to be the preeminent resource for evaluating biological safety and to build data analytics infrastructure for a large-scale efficient surveillance system. Okay, so as I mentioned, we were part of it before, and then in August we wrote a proposal as part of an RFA that had come out to be a convener. So a convener performs several functions for the FDA, and we'll go into more detail on the next slide. Some of this is administrative functions. Let's actually go straight to the next slide where we can talk about it. So Odyssey is a convener, which means that it works on several components: meetings and workshops in the seminar series, to host meetings and execute workshops for the FDA. And this is led by Mike Pilastri and David Madigan at Northeastern University, and we'll be hearing about that from David. Next, methods research development. Hold on, thanks. To develop methods for observational research for CBER, and that's led by Marc Suchard. I warn you in our presentation, so that we don't keep flipping back and forth on speakers, the methods is spread in 3 sections, so you'll see that in a moment. And then finally, we'll go over training, outreach, and engagement to educate the FDA staff and stakeholders on best methods, and that's led by Rita Kafka at Columbia. Obviously, the first one and the last one are affected by COVID, and we moved to virtual forums as necessary. Okay, next slide. So first, we're going to hear from David Madigan.
Hello. Let's see. Is this is it the next slide? This isn't my slide, right?
I think it's your slide talking about meetings and workshops, which follows, and then the next slide is about the row center.
Okay, very good. So we've, basically as a convener, we are setting up a series of meetings and workshops. They're listed here. What we're primarily focused on, the reason I was confused, in the first instance is a seminar series, which there's a sequence of speakers that are set up. We just heard about the first one will be tomorrow, Professor Salmon from Johns Hopkins. But there's a variety of meetings that are in various stages of planning as part of this work. Next slide. I'm going to take the opportunity to tell you a little bit about what's going on with regard to Odyssey at Northeastern. I moved to Northeastern about a year ago, Northeastern University, and there's a very interesting development at Northeastern, which is something called the Roux Institute. So Northeastern is based in Boston. but the university has launched an institute in Portland, Maine called the Rue Institute. It's backed by very significant philanthropic dollars. And the idea basically with the Rue Institute is to create a tech and life sciences entity in Maine that is tightly coupled with the Maine economy. So we're working very closely with partners in Maine. So there are significant resources available to basically spin up research projects, mostly in the life sciences and a couple of other areas in Portland, Maine. So as part, under the auspices of the Rue Institute, we have established an Odyssey Center. We're calling it the Odyssey Center at Rue. Website will be coming soon. And we hope that this will become a major asset to the Odyssey community. We're just beginning. We are building out a research portfolio that is related to core Odyssey activities. And right now we're very focused on developing a suite of educational programs that will culminate in probably a master's degree, but will be highly modular. So we'll have various educational offerings that will be complementary very much to the Eden Academy, and we're working closely with the Eden Academy. But badges and certificates of various kinds, we will make those available to the community and to our partners. And we also plan to provide support services to the Odyssey community. In some situations, serve as a legal entity for Odyssey if and when appropriate. And we don't yet have a website, so we kind of don't exist, but it is under development right now. Next slide, please. So the FDA, the piece of this BEST project that is at Northeastern is under the auspices of the Odyssey Center. And this is just alluded to a second ago. These are the first slate of speakers for the seminar series. Next slide, please. Just very quickly, and it's outside the scope of today's topic, but just FYI, methods work is getting underway at NU. We're in the process of hiring some people. We have a couple of faculty members involved. We think at the Odyssey Center, we will focus especially on predictive modeling and causal inference from longitudinal data. So think data that are timestamped. So think, for example, waveforms like EKGs or blood pressure readings or whatever, or just other things that are measured repeatedly over time. There are certainly methods that exist for predictive modeling and causal inference in that context, but there's very little that is at Odyssey scale. So we think this is a place where the Odyssey Center at Rue can make significant contributions. Next slide, please. Another area that we are beginning to focus on is actually a topic that I talked to. It was a community call about a year ago where I spoke about this. So this is work that George and I did that we think we can extend in various ways, which is basically, we're calling it Bayesian calibration. The basic idea is when making, in particular, a causal inference, but also potentially predictions, you want to meld together as much information as possible to bring to bear on that estimand. And there are the Bayesian framework is just very elegant and natural for accomplishing that. So they're the kinds of things that we are beginning conversations about and beginning work about at the Odyssey Center at Rue. Next slide. Actually, that's me, I think. Over to Marc.
Well, so what happened is, so thank you, David. And so we're going to hand it over to Marc. When we started this contract, we were working on the research that Marc will be presenting. And then COVID happened, and they asked us to take a break and work on something related to vaccine safety. So unfortunately, the slide that it got put in the middle, but go ahead, Jody Ann, and then we'll hand it over to Marc. So I'm just going to go a little bit over the vaccine safety, then hand it over. So just to motivate it, AstraZeneca, you've read about March 11th through 15th, 13 European countries suspend use for fears of blood clots. As of the 16th, 20 million persons were vaccinated. There were several deaths, 500 thromboembolic events, 7 with DIC and 18 with sinus thrombosis, 2 rare diseases. On the 18th, the EMA came out and said that the cost-benefit, the benefit outweighed the risks. To quote their report, those 500 events were actually lower than expected in the general population, although those rare ones, DIC and sinus thrombosis, were above baseline, but still rare. the number of events exceeds those expected, but they note the difficulty of estimating baseline incidence. Yesterday, the news came out of the good result from the vaccine in the US study, although at 2 a.m., the other news came out, whether they included all the most recent results. So the story is changing minute by minute. It's hard to keep the slides up to date. The important thing I want to point out is those bolded things. Expected in the general population exceeds those expected, difficulty establishing a baseline incidence. This is what we've been tasked with in this emergency situation by the FDA to help them with. Next slide. So we're looking at adverse events of special interest, that is vaccine-induced outcomes. And one approach is to compare what comes in with rates that are, in some sense, baseline. The European Medicines Agency, that is the US, the European counterpart to the FDA, compares reports that come in, say spontaneous reports, to overall rates for the population. The FDA has a slightly different protocol, which is using database-specific rates. That is, what's normally the rate for people in the database, and then what comes in after they're vaccinated. Odyssey Methods research involves several areas of this: phenotyping the adverse events, producing rates for agencies, along with caveats of how those rates should or shouldn't be used, the sensitivity of these rates to design choices, and this was really the initial FDA emphasis, and then comparing surveillance methods with each other. That is, using baseline rates or skipping baseline rates, just doing a self-controlled case series or our normal cohort comparison led by Martijn Schumi in the PLA-PLP workgroup. Next slide. This is part of a broader Odyssey initiative. So on the left are the people on the project that have been most focused on these vaccine safety, but on the right is the broader Odyssey group working on it that I want to highlight. And we're all working together, and sometimes it's hard to differentiate what's the FDA specific research, what's the Odyssey community research. Next slide. To point out that the EMA has its protocol, which I've already described, again, baseline rates and 23 adverse events with some controls, but again, kind of doing a population level incidence estimation. Next slide. And the FDA, on the other hand, with its incidences, looking specifically database specific, mainly in 4, and then perhaps more after that, claims databases. Also looking at EHR databases, although their protocol is now just about the claims databases, using very specific statistical models to do that. Our job is to look at the sensitivity of the baseline rates. To put it shortly, if we have these statistical models that are very fine and figuring out a difference between 1.2 and a 1.5 odds ratio, if the baseline rates are off by a factor of 1,000, we don't need to worry about the statistical test. We got a bigger problem. Next slide. The outcomes that we're studying are taken from the FDA protocol, and they're shown here, and they vary in incidence, as you'll see in a moment. Next slide. Well, so the first one, remember, I said was phenotyping, led by Patrick Ryan, looking at the literature and the protocol definitions using Odyssey tools for knowledge engineering, Odyssey tools for evaluation, and comparing between the rates we get for the FDA definitions, which are very US specific, for example, using ICD-10-CM, versus the more general ones that we use because they can run across the Odyssey network. Next slide. And then descriptive epidemiology of adverse events for COVID-19 vaccines. These are producing rates that could be used by an agency like the EMA, Shintong Li from Oxford leading this one. Basically, let's come up with the best background rates we can come up with from 2017 to 2019, looking at 200 million person years. And the incidence, as I said, varies from 1 per 100,000 person years for transverse myelitis, a very rare disease, to 1,500 for a non-hemorrhagic stroke in older patients. And I'm going to show you more results from that in a moment. Next slide. Method study related to estimating incidence rates. Anna Astropolitz from Columbia leading that one. What are the design factors? Clearly, it varies by age and sex, and there are database variations for different measurement error, but also what's your target population? What time at risk are you looking at? Anchoring, seasons, and so on. We wrote a protocol that was edited by the FDA and has been executed, and we're just producing those results now. Next slide. These are slides you saw 2 weeks ago, so I won't, this is showing the effect of age. Just note on the left-hand side, just as a reminder, That's a log scale that goes over a factor of 10,000. And those lines vary by as much as 1,000. So depending on what age group you're looking at, for the same disease, for the same database, the incidence varies by a factor of 1,000. Therefore, if you are going to compare current rates to baseline rates, you better make sure that their ages match. Next slide. And this is just showing 5 more outcomes. And then the next slide, a few more outcomes. Now let's go to the next slide. This is a new graphic that we came up with on Sunday on papers that are submitted or about to be submitted on the Incidence. Shinton Lee's first author on that. The color scale is common is orange, uncommon, a little less common is yellow, going down to green being the least common. They're sorted by the maximum incidence in any age by sex group. And we see stroke, infarction, thrombo, DVT being high on the list, transverse myelitis, Guillain-Barré. The important thing here is we never produce an incidence rate for a disease. We only produce incidence rates per age, sex, stratum. And that's like the main lesson from this paper. And then we give practical estimates that can be used. And the spread that we see within database, even after we do this age by sex strata, we get differences among databases, again, due to populations and measurement error. Next slide. And then we test hypotheses as part of the measurement study. The difference that we expected differences between male and female. The biggest effect is age. The second biggest effect is anchoring. So this is a within database problem that can't be fixed by just stratifying, which just depends on when does someone get vaccinated? When they're there for a medical visit, when they're going to a drugstore, or they're called in at work. And each of those can have a large effect on the incidence rate. And then I won't go through the others. Next slide. So our conclusions, the age effect is strong enough that it must be adjusted for when you do these analyses. Anchoring is also strong, and we're studying that further. And then databases differ. Sex differs just a little bit, not as much as the others, but we have it available. We might as well adjust for it. Our rates were actually supplied to the EMA last week in the midst of that AstraZeneca analysis. You could argue that the most important thing Odyssey has done yet is supply rates that were potentially used to make its decision about the AstraZeneca vaccine, which affects, tens of millions of people. Okay, next slide. Now I'm going to turn it over to our fearless leader, Marc Suchard, who is leading the methods analysis section, and show you what we actually are doing and intended to do from the beginning. Marc?
Thank you, George. I'm going to build upon some of the research that George just introduced that Anna and Shitong are leading in terms of developing a framework for meta-analysis or pooling estimates when it's possible for very rare events. And of course, the motivation here comes from vaccine safety work, which George just went over. Many of those outcomes are extraordinarily rare. And so if we do want to estimate a relative safety incidence for a safety event or incidence rates themselves, one direction you might go is to think about using very large populations or pooling information across databases or increasing your follow-up time. Unfortunately, there's a very pressing need for answers now, so increasing follow-up time is not a very viable direction. One of the challenges that we have is that many of our data sources in the Odyssey network are federated. And what this means is we have very limited ability to share, if any, individual patient data. So the best that we can do in developing large populations is not by sharing the individual patient records, but summary statistics about them across different databases. Unfortunately, there are some real current limitations on how standard meta-analysis works that precludes its application for very rare events. And the thing that you need to keep in your back of your mind is most standard meta-analysis relies on very simple summary statistics. And the idea that these summary statistics, a point estimate and a standard deviation, completely describe the uncertainty in the risk estimate that we're trying to pool from site to site. Basically, it means you need to assume asymptotic normality. I like to talk about sort of the realm of asymptopia. This is where asymptotics hold. And when we talk about vaccine and rare safety events, we're far from this mythical land of asymptopia. Next slide. So let me give a brief review of how our current strategy works in distributed research network and how we're going to modify this approach to help effectively pool estimates for rare events. So under our current strategy, any site can lead a study. So this is Site A. Site A, or Anna or Shitong, will develop the analysis code locally. In the next slide. And then this code is then distributed out to each of the individual study participants, so different data partners within the Odyssey network. After that, results are then generated within the federated data sets, and aggregate statistics are then sent back to Site A, or the lead study. Next slide. And next slide. An example of this success is a paper that the community wrote now about 4.5 years ago, looking at the relative risk of angioedema for comparing individuals who are taking Keppra versus phenytoin. And this was motivated by a drug safety release from the FDA. And we asked ourselves, can we respond to this FAERS notification in a timely fashion? Next slide. When doing that, we were able to recruit 10 different data partners, but a number of these data partners actually in their data set came up with no angioedema or the adverse event that we were looking for. So of the 10, we really only got point estimates. Here it lists from 7 different data sets, but you'll notice like IMS ambulatory, that point estimate really doesn't mean anything, right? So they're really only, out of the 10, we could only use data from 5 of those to create our composite summary statistic, which showed no increased risk for angioedema. Next slide. So what is the information that we're pulling back? It could be a relative risk ratio or a hazard ratio, which I'll describe here. And we get back a point estimate and its normal and its standard error. And based on that standard error, we assume it's normally distributed around that. Move on to the next bits. You can actually just fill out the whole page for me, Jody Ann. Sorry about that.
That's okay.
Which includes kind of the optimum, the standard error, and that gives us sort of an uncertainty region about which we think that the true result holds. The trouble is when the number of outcomes is relatively rare in any of the target or comparator groups, like in this case, there were only 3 outcomes, the true shape of this function is far from normally distributed. Asymptotics really don't hold. Let's go on to the next slide. So if we assume, so here's a real world example using a very large database. Here we're looking at the relative risk of a rare outcome for individuals taking 2 very popular antidepressants in one of the largest claims databases around, CCAE. Well, this is across a number of different databases. And if we assume asymptotic normality, we can create a summary statistic which sort of covers 1, saying we don't have strong evidence to reject the null hypothesis. However, let's go on to the next slide. Sorry, the asymptotic normality does slightly reject that null hypothesis. However, if we make no assumptions, asymptotic assumptions, and instead of share the shape of the data information, we actually find that our estimate clearly covers the null hypothesis, and we don't have evidence for a difference here. So, so relaxing the asymptotic normality assumption is critically important when we're talking about rare events if we want to be returning reliable evidence. Next slide. Why does this happen? Well, for many of the estimates that we're interested in, like the hazard ratio, comparing the relative risk of a rare outcome, or the incidence rate ratio, is not normally distributed. Here's what the likelihood function actually looks like in a real data set where there are no outcomes in one of the two comparison groups. And it would be really hard to say that there's a point estimate, here it doesn't even exist, and what that information would entail. So traditionally, what would we have to do? We just throw all of that data out, as we had to do in the Kepra study. So here's an alternative solution. Next slide, please. Instead of just returning a point estimate, which may or may not exist for many of these rate ratios or hazard ratios, we're actually going to return from each of the data sets the shape of the likelihood function. And then we're going to apply Bayesian inference machinery to combine these shapes in a total evidence approach that extracts as much information about these rare events across multiple data sets simultaneously. And this is enabled by a new Markov chain Monte Carlo package within the Odyssey community that allows us to do high performance computing for large scale Bayesian models. Next slide. So sort of here's a graphical view of sort of the total evidence approach, which will give you the take-home perspective. When there's in the middle on the top, there's a relatively informative data source, but it's asymptotic. But it's asymmetric, it's not asymptotic. We can include that information, that we have more information in one shoulder than in the other. In addition, in sort of data limited resources with rare events, where events may not have occurred in some of the different age strata that are so important, then we do get lower bound or upper bound information. And instead of throwing that away, we can simultaneously pull it together to get the total evidence across all data sources. So let's move on to the next slide. So where are we with this work? Well, this is joint work with Martijn Schumi, George, Chen, and David. And what we're currently working on at the moment are thinking of sort of optimal ways of communicating this likelihood function and comparing their performance for both drug safety and vaccine safety examples. If you're interested, there's an archive manuscript. And it gives us a foothold to start thinking about what are the next steps. A particularly relevant question that the FDA raised with us in the last couple of weeks is, okay, these are good for models of hazard ratios, comparative rates, but what if we're interested in kind of, incidence rates with very small counts in vaccines where we say stratified by age, which is critically important. In some of those age categories, no events are going to happen. And if I want a relative risk, then how do I avoid dividing a 0 rate by a 0 rate? And sort of the punchline solution that we'll provide is these are easy to turn around in terms of Bayesian inference questions, which allow us to, take a total evidence approach and return a relative risk estimate when we only have data from one shoulder or the other. So if these types of problems are of interest to you, please reach out to us. Again, we're an open community, and we're happy to engage with other researchers. So thank you for the time, George.
Okay, now we have Rita Kukafka on training outreach and engagement. Rita?
Hi, could you hear me? I'm just unmuted. I want to make sure.
Yes.
Okay, good. So I'm Rita Kukafka. I'm Columbia University, and I'm going to talk about the training outreach and engagement that is part of the FDA contract that we've been talking about. So this is just a very high level. We want to conduct training, outreach, and engagement of FDA staff and best stakeholder community to improve and advance BEST as a system that meets the needs of the FDA and the community. So that's the overall goal of this part of the contract. I'm going to just talk a little bit about where we are and some of the things that we're ready to embark on. I should mention we were put on hold because of the vaccines and the role of the FDA in the vaccine approval. So this is actually first getting started. And the way we're going to begin to this goal to meet this goal is we want to first identify, we want to characterize We want to gather stakeholder input and information. And this is what might be familiar as a needs assessment. And the methods we're going to use are going to be interviews, surveys, meetings, and workshops, again, depending on what we can't do during this pandemic, but we will gather this information. The purpose of collecting the information is to inform approaches to engage the stakeholder community and foster and encourage the use of this infrastructure for safety and effectiveness evaluations and surveillance, real-world evidence generation, and other related activities. So it's relatively broad and very consistent with Odyssey goals. Next slide. So I do want to just get into a little bit of the nuance. We in terms of how we propose to implement this training, this will be for targeted, and what we want to do, and I and Columbia have had a good amount of experience doing this. And what we aim to achieve is to really stratify our audience. We want to define the audience, and then we want to target different groups who may vary in their knowledge and understanding. So again, it's not, the idea of this is not the one size fits all, but really to understand what the needs are, and then to be able to target the content that we do develop appropriately to that audience, recognizing that some groups, maybe all groups, would want this high-level presentation of concepts, whereas others, a more stratified group of individuals, might be interested in different kinds of technical sessions, for example, for epidemiologists, clinicians, and informaticians, and scientists. So we really do want to do this needs assessment and then stratify and then target. Next slide. In terms of how we propose to deliver, we are planning, and we've had some initial conversations with EDM Academy as the platform for delivering the training. Again, depending on what we can and can't do over the next, hopefully we will in a couple of years be able to do more in person, and certainly we want to, even with in person, we do want to continue to train virtually. We think it's just more convenient, it's easier, we reach more. We have a similar kind of project that we've worked on for the for training technology, and we've got about 15,000 people already enrolled, so we could reach large numbers virtually. And the other thing is, with virtual, you do it once and you could really disseminate it often. And as I said, we do have experience in this, and one of the methods that we did employ previously is this case-based format. And the goal here of the case, I don't want to get too much into what that is, but the important part of this is that it's designed to really engage into real-world applications. So it's really like developing stories and then inputting the content that we deem to be appropriate for different stratified groups of the population, and deliver it that way. And we have been able to do that in the digital environment. Next slide. So I'm not going to spend, again, too much time, but this is pretty much the methodology that we draw upon. It's called design research methodology. And what's interesting about this methodology is very, it's the most unique part of it is the stakeholder engagement. So we don't miss the mark. We understand what the characteristics of the trainees, how the population segments, and what topics are most important. So it's this cycle of assessment, evaluation, and it makes good use of the technology platform in which we plan to implement. Next. In terms of, this is a little bit more about process. So what we do is, once we do the needs assessment, which we're about to start doing now, is we develop competencies. So we don't just hit topics, we really define competencies, and we do a match between those competencies and the different types of trainees and subtypes and the needs of those subtypes. We involve, and this is actually a call for engagement if anyone has interest, we really do rely on subject matter experts. So subject matter experts who really understand one topic very deeply are very, very important to this process. and they really do help us with developing those competencies and being able to evaluate the training. As part of the evaluation, we also plan to track numbers of trainees, types of training, and also provide a training assessment. I think so this is my last slide, and this is again a call for anyone who wants to become involved. We are in this current year, again, we've been delayed because of the FDA's role in the vaccines, but we're now about to begin the task of training and outreach by conducting these formative methods, both surveys and qualitative interviews. So we're just ready to hit the road, and my name and email are up there. Please contact me if this is something you might be interested in. All comers are welcome. Thank you.
Thank you, Rita. Let me just take a moment to recognize Jody Ann McLagan's role as program manager for this project with FDA. So thank you, Jody Ann, and she's the one who's been running the slides quietly and well in the background. And let me remind everyone once again, tomorrow's talk with Daniel Salmon. Let me turn it over to Craig and Patrick to run the Q&A.
Great, that was a wonderful presentation. So thank you again to George, David, Marc, Rita, and Jody Ann for all the work that you put into this. We will open this up to a Q&A right now. So if you want to raise your hand, we'll try to call on as many people as possible. I know there was something in the chat. Patrick, did you want to hit on that?
Yeah, thanks, Craig. So when Marc was talking about concerns about small counts, Sammy Sweeza in the chat posted about use of rate differences instead of hazard ratios in the presence of zero. So Sammy, if you are able to unmute, maybe you'd like to talk a little bit about that, and Marc, maybe you could respond.
Yeah, hi. Thank you, Patrick and Marc, for the excellent presentations. I just remember from many years ago issues about meta-analysis where there were tons of zeros, and then some papers were written about why not use the rate difference. And I saw your examples here. A hazard ratio doesn't mean very much when you have 3 events in one group and 1 event in the other group. The time to event doesn't bring much information. So all of the information is in the number of events, and then if you have zeros, and even if you have 0, 0, I think you can get some information from the sample size of the 0, 0 by using a rate difference. And again, I'm not knocking the Bayesian approaches, but I think when you want to publish in clinical journals, simpler is usually better and easier and more understood. That was my point. But I think there is a literature out there. And thanks a lot again, Marc and Patrick and everybody for doing this.
Yeah, no, Sammy, those are some wonderful and very important comments. And to start off the big picture, I completely agree with if there are good approaches that are simple, we should be exploiting them. And in terms of the difference, I think it's an important place to look, right? Because even with 0 events, we have a bound on what we think the rate would be, and you can look at the differences between bounds, which are much more informative than the ratio of different bounds, and we can be reporting those quantities without having to pull in a large amount of extra machinery. An important question that someone in the community might want to explore is what are the performances? What are the performance characteristics of taking a full Bayesian approach, which in many cases may not be necessary, versus reporting the difference in the bounded rates? And I'd like to invite people. Actually, I already got a message. I'm going to call out Adam. You already sent me a message saying, how can I get involved with this? I think that's a great project to look at. So thank you, Sammy.
Other questions? There's no hand raised at this point, so maybe some people don't have that option. So if somebody wants to just kind of fire off a question, unmute yourself.
Rita, I've got a question for you, because you mentioned multiple times the lament of being virtual right now. And I know we've all been sitting on all of these Zoom calls all of this time, and certainly share the feeling that it sure will be nice when we're able to be in a classroom together. I'm talking about this. Could you just? But one of the things you commented is we're looking to use the Eden platform and other things, and of course we are a virtual community. Could you just comment a little bit about the value of the in-person training experience and what your experience has been in being able to get together and collaborate in person, and also what the trade-offs are versus the kind of virtual experience that we've all kind of had to succumb to lately?
Yeah, thank you, Patrick. So I have to say that my work with virtual training and using, a platform like EDUM, and I look forward to using that platform, is that happened before COVID. So that wasn't the default. That wasn't the lesser. So there are advantages and disadvantages for both. I think that we've certainly been involved in more traditional classroom training, and there's definitely advantages to that. But we have learned, and I've seen success, and I mentioned these case-based methods that really do manage to engage in a meaningful way, even when it is not in person. The other advantage of virtual is that reach, and anytime, any day, doesn't matter what hour, you're really working on your own pace. I do, of course, there are benefits to the in-person, would look forward to that as well, but I wouldn't, again, the experience with virtual was pre-COVID. Maybe we're going to appreciate in person more than we did, but they're good companions, put it that way.
Rita, Patrick, can I chime in? This is Sami Suisa again. With respect to teaching, I just want to share our experience at McGill. We have a pharmacoepidemiology summer program with 6 courses in this summer program. One of the 6 courses is critical review of the literature of papers. So we take papers, we have a critical review. It is restricted to 20 students, and it is always in class. It has always been in class. But this is one course that we have not been able to make into a Zoom course. All of the others are now done in Zoom because of what's happening. But that one course has been postponed to next year for the second time because it is impossible not to do live with the interaction that you have, the live interaction rather than the virtual interaction. It has just been impossible. So just a sharing that one of the 6 courses we can't do.
Yeah, that's very interesting, Sammy. One of the Odyssey tutorials exercises that we've done in our prior trainings has usually been a small group session where we give people one of the papers that was published actually by our colleagues at the FDA. It's a very good paper, but we ask people to read it and critically evaluate, like, do you have all the information to replicate it? And there's something magical about sitting at a table with 3 or 4 people digging through the same paper and underlining and highlighting that is much more difficult. Like, you don't get the same impact when you just, talk to it, or if you have, the hands up coming in the Zoom. There's something about, forcing people to argue with each other at a table and having the ability to slam your fist down that conveys some extra experience for the trainees.
Yeah. I think there's like the speed of interaction. There are some things that it's just too hard to get it over Zoom because you can't communicate fast enough. Second is the parallel interaction where people are subtly pairing up in different ways. And the third is the nonverbal communication that happens that you can quickly tell that the professor's not happy with what you're saying and steer clear or steer towards something that you just can't tell on Zoom, and it just takes too long, and then you've forgotten what you're talking about.
I want to come back in the defense of what, my experience has been. And I have to say, it was not on Zoom. So I would agree that some of the things that, you're talking about cannot happen on Zoom. But in a different kind of platform, and that's why I'm interested in continuing discussions with the EDUM Academy, because we were able to, what worked for us is this idea of problem solving. So you'd sort of build in that, again, it's not in person, but it's just not Zoom. So I guess my point is there's somewhere in between Zoom and in person.
Yeah, I mean, how do you think all drug companies that are multinational develop drugs?
Exactly, yeah.
You know, you're doing huge submissions and you have to have meetings. When a drug company I was with, we were in US, you got Europe, you got Australia, you know, you have to learn to deal. And I get it that collaboration is important. This is somewhat, you know, gone on a bit longer. There's no travel and stuff. So, you know.
If I may, it's Nigel Hughes here. Hi, just regarding the academy. I think a key emphasis on how we've structured the academy to date has been very much focused, and as you outlined, Rita, on adult-directed learning. And in fact, the vast majority of what we provide in the academy is for individuals to spend time when it fits them best and within their work plan and some personal time to do courses and come to badges and so forth. But you know, with that kind of flexibility. But certainly, you know, that's a complement, I think, again, to working within groups or similar, or in class environment or whatever. So I definitely agree with you. You know, the best we have is a complement of all. But currently, clearly, with the restrictions, that remains, and sadly at the moment still remains a bit of a challenge. Absolutely. So, in the absence of anything, I still think we have great assets with the Academy, the Book of Odyssey, and various other resources that are available. But you know, shall we say, more traditional classroom-based didactic lectures and symposia and so forth is something I think majority is still looking forward to, particularly as it enables, you know, going from the individual to the group, it enables that ability to work as a group, to think beyond you as an individual, and also to help innovate your learning, but also in terms of the outputs. A bit like the experience we have with study-a-thons, of course, as well as partly an educational experience too.
About 2 minutes. I don't know if there's any final thoughts or questions.
So Marc and George, I'd like to ask you guys maybe a provocative question here. We're collaborating with our colleagues at FDA CBER, and they've presented some interesting and different clinical problems that they have to face on a daily basis. And we've largely have come from our vantage points and have worked on other types of problems, but some of the new problems that George, you alluded to with, you know, trying to dive into blood products, or now many of us like throwing ourselves into thinking critically about vaccine surveillance. So I'm curious from your perspective. Obviously, we're trying to help our colleagues at the FDA and EMA by contributing our insights, but I think we're also learning a lot along this process. So maybe could you highlight some of the areas where there are kind of new or different problems that you're learning from and are excited to be trying to tackle now that we're in this collaboration with the FDA?
Sure. I mean, I'll start and then hand it over to you, Marc. Is that?
Well, I was going to say that's a great question. I'm sure George can have a start at that.
So first of all, phenotyping. We're going into an area with new kinds of data, new kinds of problems that we haven't been. So like transfusion being a perfect example, with new challenges. So I think, and there may be adjustments to our tools and our coding. You know, they're all parts of our process may be affected by trying to capture problems. Or just the fact that a vaccine was given, the fact that a transfusion was given. Transfusion medicine brings in shorter timescales. Things are happening on seconds, minutes, hours, and not as much on months and years as we might with outpatient, say, hypertension, antihypertension drugs. So a lot on the phenotyping that then affects, you know, the temporal side, then affects the analysis. David Madigan alluded to the longitudinal time series earlier in the talk. So I think that's very important here. And then the third thing I think of just utility, like we getting like, what is actually useful? So we're working with the FDA, and the FDA team they're working with is very smart and very, you know, good at this. We're trying to add value for them, and we're trying to seek out, well, what is it? What gaps can we fill or augment or whatever? And so it's kind of a new world for us a little bit.
Yeah, I mean, it's an amazing group, and it's a fantastic opportunity to kind of look behind, look behind the wall and see that there's great work going on in the what great work is going on in the regulatory agencies, and how that intersects with what we've been developing in the Odyssey community. I think one of the large gaps between what's traditionally gone on in the FDA and the Odyssey community is the idea of network open research. And I think we as a community have a lot to learn from the FDA and how we can use a community, an open community, to help inform regulatory questions in a way that have utility, and are easy and straightforward to present. Kind of harking back directly to Sammy's statement, sometimes it's easy to take the very simple approach to a question. The challenge is the simple approach might not be available for us as we move into this open community, large data setting. And so there's very much a two-way street in education, and I'm glad that Rita is on board to help us both get educated and be better educators for this two-way street.
All right, we are a little over the hour. So again, thank you to George, David, Marc, Rita, and Jody Ann for this great presentation. All recordings will be on the community calls page later today, as well as the links I mentioned earlier. Thank you again for joining, and get those visualizations posted. Thanks, everybody.